Hello, everyone. I'm going to discuss cyanide poisoning, an important topic that illustrates concepts we have covered in proteins and metabolism. Cyanide is a chemical compound that contains a cyano group where carbon and nitrogen atoms are triple bonded together. Cyanide is a very potent ligand for many transition metals. A ligand is an ion or molecule attached to a metal atom by coordinate bonding. High affinities of metals for cyanide relate to its negative charge, compactness, and ability to engage in pi bonding, which is a covalent bond formed between two neighboring atoms' unbonded p orbitals. Hydrogen cyanide was discovered in 1782 by Swedish chemist Carl Wilhelm Scheele, who prepared it from a dye used for the uniforms of the Prussian army. Cyanide, as a poison, dates back thousands of years to the Roman Empire when Nero used a naturally occurring form to poison his enemies. Cyanide-containing compounds are used for many chemical processes, such as case hardening of iron and steel, electroplating, and photograph development. Cyanide has been used as a poison or chemical warfare agent, but most exposures are unintentional. House fires are the #1 cause of cyanide poisoning in Canada. Cyanide is released during the combustion of products containing both carbon and nitrogen, such as wool, silk, and polyurethane. Significant levels of cyanide are present in 35% of all fire victims. The toxic effects of cyanide result from inactivation of cytochrome oxidase and inhibition of cellular respiration, and consequent histotoxic anoxia, the inability of tissues to use oxygen delivered. Cyanide binds to Fe3+ in cytochrome A3. As a result, O2 cannot receive electrons. Therefore, respiration is inhibited, energy production is halted, and cell death occurs rapidly. The body handles small amounts of cyanide differently than large amounts. In small doses, cyanide is changed into thiocyanate, which is less harmful and excreted in urine. In small amounts, it can combine with hydroxycobalamin to form vitamin B12, which helps maintain healthy nerve and red blood cells. By stopping oxidative phosphorylation and its ATP production, intracellular oxygen utilization ceases, and cells die. Cardiorespiratory and central nervous systems are most susceptible to cyanide poisoning because of their high dependency on blood oxygen supply. Unable to use oxygen, cells are then forced into anaerobic metabolism, which creates lactic acid and leads to acid-base imbalances, specifically metabolic acidosis. The electron transport chain is a site of oxidative phosphorylation. The transfer of electrons from NADH and FADH2, supplied by earlier steps of respiration, to oxygen takes place via a series of electron carriers. This is catalyzed by the cytochrome oxidase enzyme system in the mitochondria, and the impairment arises from the inhibition by cyanide of the enzyme cytochrome C oxidase A3, which is found in the inner mitochondrial membrane of eukaryotic cells. This occurs due to the high binding affinity of cyanide for the ferric ion found in the heme molecule of the oxidized form of this enzyme. When cyanide strongly attaches to the iron within this protein, it results in loss of structural integrity, shape, and, therefore, effectiveness of the enzyme. Thus, the ETC is disrupted, meaning that the cell can no longer aerobically produce ATP for energy and will die. In normal functioning, the electron transport chain uses the shuttling of electrons to transport hydrogen ions into the intermembrane space. The ions are returned to the matrix via ATP synthase, and the electrons are removed from the chain by oxygen. Cyanide binds non-competitively to cytochrome oxidase, complex 4 in the diagram, and physically changes the shape of its active site such that electrons can't be released to oxygen and the ETC halts. Cyanide is a non-competitive inhibitor. It binds to an allosteric site on cytochrome oxidase. By changing the shape of the active site, functionality of the enzyme is compromised, and cytochrome oxidase can no longer pass electrons to the final acceptor, oxygen. the ETC can't function and ATP is not produced. In minutes, short-term exposures to low levels of cyanide via inhalation, skin absorption, or ingestion can lead to rapid breathing and heart rate, restlessness, dizziness, weakness, headache, and vomiting. Moderate inhalation exposures can add confusion, anxiety, and shortness of breath. Acute exposure to high cyanide concentrations lead to death in minutes. Long-term exposure to low levels of cyanide can lead to breathing difficulties, chest and heart pain, vomiting, loss of appetite, headaches, nosebleeds, and damage to the brain and heart. Larger amounts of cyanide can cause rapid convulsions, lowering of heart rate and blood pressure, loss of consciousness, and respiratory failure that leads to death. First aid treatment of cyanide poisoning. Speed is essential. Seek immediate medical attention. A doctor may administer one of two antidotes, cyanide antidote kit or hydroxycobalamin cyanokit. The cyanide antidote kit consists of 3 medications given together, amyl nitrite, sodium nitrite, and sodium thiosulfate. Hydroxycobalamin detoxifies cyanide by binding with it to produce non-toxic vitamin B12. This medication neutralizes cyanide at a slow enough rate to allow the liver enzyme rhodinase to further detoxify cyanide. Cyanide antidotes are classified into 4 major groups on their mechanism of action: scavengers, detoxification, physiological, and biochemical.